It's not clear that the evolution of the biosphere is a proper topic to me. So I changed the question. That is, what are the ingredients that any planetary body needs to encourage life to develop? How does life alter the planet so the planet could be recognized as alive? And how do the spheres, the atmo, litho, and biosphere co-evolve? These questions seem that they're at least somewhat tractable. But after all, I've heard about the variations in planets from the last day. They may be tractable, but not today. Okay, this is extremely difficult and challenging stuff, and all the more reason to attack it. But it's a challenge. Okay, so I'm going to first deal. And by the way, I'm going to be a little bit speculative here and probably say some things that you might wonder what I'm talking about or disagree, and please raise your hand and ask me. I think it's more appropriate while we're trudging through this to address any questions. What the ingredients are, and I have to say also, I'm a microbial chauvinist. If you don't like it, it's too bad, because that's, I've been a microbiologist for 45 years, and I ain't gonna stop now, okay? But so when I talk about the evolution of the biosphere, for the most part, I'm talking about the first 1.5 billion years of life on the planet, which I think is appropriate with regard to exoplanets, and we can talk about why that is. But what the ingredients are pretty simple from my point of view. We need some energy. That can be a nearby sun. It's kind of the essence of the habitable zone idea, and the zone where liquid water is possible. When I look at our own solar system and see the expeditions to Europa and Titan, I'm not sure that the habitable zone has much of a meaning for any of us, but we keep talking about it. Okay, what I would say is even on those far off places, the radiant sun is still an important component. The other one, if I put my geobiology hat on, is the hot core, which is a ready supply of thermally reduced substrates for life. Hydrogen, hydrogen sulfide, iron, you name them, they're coming up in the hydrothermal vents. They're places that are continuously generated. And from the microbiology point of view, it's a buffet dinner, okay? So this also brings up the interesting role of plate tectonics that we heard about yesterday and the heat flow of the planet, which I think must be extremely important for this kind of thing. Now, fuels and oxidants are things that I think about all the time, and Diane did a great job of introducing this. We saw it yesterday. I like to look over Stuhm and Morgan. If you don't know this book, you should know it. It's a great aquatic chemistry book. And they deal with talking about the fuels and the oxidants of the world. And a little bit later, I'm going to compare what a simple chemistry cycle of some of these things would look like as opposed to a biological cycle. Okay, and that's what I want to deal with. But the fuels of life, if we look at the microbes, are pretty much everything we can see that will give up an electron. Some microbe has figured out how to take that electron. Okay, so us eukaryotes are pretty fussy about what we use for fuels, and they're all in this category. The rest of the prokaryotic world not only uses organics, but it uses all of these fuels here. The oxidants of life on the same scale, by the way, this is the PE adjusted to water at pH 7 for this scale here, which is why I like Stuhm and Morgan for a biologist. And the difference between the potential here and any of these electron acceptors is the amount of voltage you can get out of this couple for biological life. It's what we use for charging that membrane or the biological capacitor that we heard about from Diane. And I like this diagram because if you take almost anything that's over here and draw an arrow to some other oxidant, and the slope of that arrow goes down, there will be some microbe that knows how to make a living by doing that. Okay, and that's absolutely true. Every one of these electron couples over here, in this case, it could be a methanogen. It could also be an acetogen, okay, that would use hydrogen as the electron donor and CO2 as the electron acceptor. Over here is what the rest of us eukaryotes use. And in this case, they're all aerobes, and there's everything between. You can draw the arrows, you can have the fun, but when we go to an environment to study the environmental microbiology, the thing we do, first thing we do is a survey. That doesn't tell you what's going on, it tells you what could be going on there. And if some of these are missing, you shouldn't waste your time studying them. Okay, this is, and of course, that's what you should do if you go to another planet. You should try to get an idea of what the sources of energy are and what are there. There are some traps in this approach, and that is if some of these are being used very rapidly, they could be the things that are limiting energy, and you won't see them. Okay, it's if we grow things in a chemostat, the chemostat's always limited for something, and in this case, that something is what's limiting the rate of growth, and we won't be able to see it by measuring the water. So this is just part of the theory here. But it's important that the geological environment, the lithosphere, is rich in many, many of these oxidants and reductants. And that's another thing you have to know about. You want to know what's there before you take a stab at what's limiting life. Okay, and the last thing is that some of these electron acceptors, notably the things that I've been working on for 30 years, are insoluble. And that includes the iron oxides and the manganese oxides. And those insoluble oxides were judged to be impossible for bacteria to use as electron acceptors. And the reason is the way that bacteria are made. You saw Diane's diagram today with a membrane where all the electron flow occurs. And so oxygen can get into the cell, nitrate can get in, all these others can get in, CO2 can get in through the membrane, and electron flow can occur down the membrane. But iron and manganese are rocks, and they're sitting on the outside, and this was deemed to be impossible in the textbooks a couple decades ago. So this leads us to the interesting story then that, oh, first I want to go, just one of the things, when I used to teach my geobiology course at Caltech, I would ask the students, could they dream up another organism that could use a different form of energy? It's a great thing if you want to have fun. You could use radiant energy with other wavelengths, right? Maybe infrared, maybe UV, maybe some other kind of electromagnetic energy. Can you use magnetic energy? I got great diagrams of organisms from students figuring out how to do this, okay? What about kinetic energy? Could you use a bacterial propeller and run it the wrong direction and make energy? It takes energy to run the propeller. You can do these. What about thermal energy? I got diagrams of organisms with long thermocouples on the end of them. And we actually see stuff like that out in nature. You don't know what it's made of, but why limit ourselves to what we only know on this planet, okay? And the last one was electric, which I always used to laugh about. And I've been working on electric bacteria now for 20 years. And we know that we have bacteria that will convert organic carbon to electricity. We know that we have bacteria that will convert electricity to organic carbon. Absurd, you say. You just heard Bob talk about bacteria that'll put photons to organic carbon. And I studied for about 10 years bacteria that convert organic carbon to photons, bioluminescent bacteria. It isn't absurd. The question is, will your mind be open enough to look at how this happens? 30 years later, we know how this is done. The bacteria make specific cytochromes with 10 heme groups on them that have enough length to them to go across the cell membrane. And instead of using the soluble manganese in the cell, they just send the electrons to the outside. So they flow down the membrane, go out to the solid substrates, and it opens up a whole window of metabolism that we didn't know about 20 years ago, okay? And this, I would say, well, I just threw in a couple of references, and I apologize, I was in such a hurry. These two references here were just published last year, and I can give the references to you if you want them. But there are probably 100 references in the literature now to these electric bacteria, and it's a part of life that we just didn't know about before. Most microbiologists have no idea of electrochemistry, so we weren't trained that way. That's why we have meetings like this, okay? So, and this is an organism I isolated called Schuonella. It will grow on iron or manganese as its energy source, and it's what we call an electrogen. These are organisms from Victoria Orphan's lab that will take up electrons from solid energy sources, and we call them electrotrophs. One of the references that I had here was the description of electrotrophic bacteria. These have some very interesting properties that because they are able to do this, you can just grow them on an electrode. And a lot of them won't grow other places. You know, one of the big problems in microbiology is most of the bugs we work with can't be cultured, okay? We have whole groups of organisms that will grow just fine on electrodes, but we can't culture them in standard bacterial medium. This is an important perspective in terms of thinking about life other places, but especially an important one for the sediments and things. So the energy, I think, is taken care of here. I would say that the other thing that I'm most concerned with is not that it have water necessarily, but it have some kind of liquid, okay? I guess maybe I'm a chemical chauvinist, but I don't see how you do chemistry of life in the solid state, okay? Somehow you need a liquid, or the great advantage that life has is going to be hard to express. And that is that life works by changing the rates of reactions, okay? It's the catalyst. You've heard it over and over at this meeting. Life invents catalysts, and those catalysts do things that regular chemistry doesn't do because of activation energies, of other reasons for why it is. And I just don't see how to do that in the solid state, okay? So and then I want a brief discussion of kinetics from my point of view, and that is that thinking about chemistry, and we go back all the way to the origin of life here, that slow chemistry is what you want, okay? Life's going to make inventions, and if you have a reaction, whoops, what did I do here? With really high activation energy, then it's pretty easy to invent a catalyst that will overcome that activation energy and speed it up. The greatest example, I think, that's common everywhere is sulfate reduction. We always put it on the map and say, well, this is what's going to happen. But the ocean's full of sulfate, and it doesn't do anything until a bacteria comes along. In order to get sulfate reduction by chemistry, you pretty much have to heat it up to about 200 degrees centigrade. Okay, so the stuff coming out of the hydrothermal vents has been reduced to sulfide. But the sulfate in the ocean is a great reservoir of electron acceptor. The other side is that fast chemistry is tough for life. If you have something that decays in nanoseconds or microseconds, it's hard to make an enzyme that speeds that up because most enzymes operate at the millisecond level. So this is what protein chemistry does. So you want to have natural reactions where the kinetics are slow. There's nothing special or hard about this. It's just that we don't think about it often. Okay. So again, with the kinetics, you want some energy sources that favor slow kinetics. You want to give life time to invent the catalysts. And it'll also invent ways to store energy that's stable. Okay, I think one of the great inventions of life are organic polymers, carbohydrates and things that you can sit on the shelf for a year and they're still there. But you give the bacteria a little bit of chance and they're gone the next day. Okay, this is storage products are one of the things. And I just point out that in some cases, because the way the Earth changed after the great oxidation event, iron too got almost completely removed from the ocean. But bacteria had the sense to make some fantastic chelators called siderophores that bind that iron to iron 3 after it's oxidized and keep it from forming an oxide. So it's a kinetic effect, but it's a negative kinetic effect. It stops a reaction. It keeps the iron up in the upper parts of the ocean bound as an organic. Important stuff, okay? So I want to say another thing about interdisciplinary work, and that is that lots of things have been discovered because geologists tell us that things are happening that we wouldn't believe normally, okay? A great example is what Diane mentioned about anaerobic methane oxidation, something that the microbiologists absolutely said couldn't be true because they hadn't found an organism that would do it, okay? The geochemistry said it's happening all over the world in these anaerobic environments, and finally the microbiologists figured it out. Another one is anaerobic ammonia oxidation, or so-called ANAMOX. And the third one is this stuff I was talking about before, and I just want to show you something if I can. If you don't believe in kinetics, you will after you see this movie, okay? So this is an organism I isolated called Schuonella. You see it swimming around here. It's in an anaerobic flat capillary, and the bugs are swimming because there's still oxygen there. And then pretty soon the oxygen goes away, and the only thing left to breathe is this chunk of manganese oxide. You can see it over there better, I think. And this is a time-lapse photo. There's 6 hours. Here's a piece of manganese with the bacteria swarming around it. Move away, and the bacteria aren't swimming at all because there's no energy. These bacteria are breathing this rock. They swim up, dump their electrons, and then swim away again. And this is happening all over the world in sediments. Whoops, come back, come back. Oh, did I goof that up? Well, you lucky folks, you get to see this again. Okay, so anyway, I'll see if I can move this down here a little bit. Here we go to the exciting middle of the movie here. So anyway, the bacteria are swimming around here. They're interacting with this. It's important that they don't necessarily form a biofilm. They keep swimming around the particle. And when all the manganese is gone, all they do is Brownian motion. They can't swim anymore, okay? This is it. All the energy in that environment is dependent on that solid electron acceptor, and these guys are just vibrating. So, the reason I wanted to show you that is that according to Stum and Morgan, that chunk of manganese oxide should have been stable for at least 1,000 years under those conditions. Okay, this is what life does. It invents catalysts that do things that are completely off scale. Okay, and this is happening in this lake in upstate New York. This was 1,000 times faster than the rate should have been. That's one of the kind of things you want to look for, okay? So how does life alter the planet? You just saw one of the ways. It does things at rates that are so fast that they outdo diffusion and you make gradients. And when you make gradients, often there will be a deposition of that gradient that is evidence that it was there. It's a layered community, it's all kinds of things. They use up these energy sources. They use the available electron acceptors, and you end up with basically the proof that life did this. And this is the kind of thing I'm talking about. You're not looking for a chemical biosignature necessarily. You're looking at the evidence that something kinetically out of bounds happened. And I think they're all over the place with different kinds of bacteria. Eventually, organics begin to accumulate of one kind or another, and the oxidized products of those redox reactions often accumulate too. And if you know what you're looking for, or better yet, if you see this stuff, you can infer what happened. So, the notable events, in my opinion, iron was adopted as the major electron acceptor and carrier, probably because the ocean was full of iron. Okay. In a way, life then played a nasty trick on itself by inventing oxygenic photosynthesis. Okay, because but iron was invented, probably electron transport was invented for respiration. Other electron transporters were invented. Those of you who study metabolism know this, but the big problem in all of this is that electrons aren't soluble. And when you start to take them off of a substrate, you better have something that's going to carry them somewhere. So you had to invent things like flavins and quinones and pyridine nucleotides to carry these electrons around in the cell. I think that's why bacteria are small. It's because the diffusion of a molecule from one end to the other takes about a millisecond. If you made bacteria 10 microns, they'd have a hell of a time with the metabolism. Okay, because they need to move these electrons around at the rate that an enzyme works. And that's, again, is a millisecond kind of scale. And after all of this, then I think you could have biosphere evolution. Okay, before this, it was organismal evolution. And now you want to talk about communities. And as you develop this metabolic diversity, the waste of one bug becomes the substrate of another. And we know how that works, okay? And I think, for the most part, microbial communities began to emerge, and you still see this in the Vinogradsky columns that you saw today in Diane's lecture. And for the most part, microbial communities are collaborating, not fighting with each other, okay? But you see this even if you slow down, use less substrate, the bacteria can adapt to this very easily. But they don't adapt. There, as far as I know, there are only 2 examples of bacterial predators, i.e. a bacteria that eats another bacteria, okay, in the entire microbial world. Okay, now there are viruses, and they will kill bacteria, but they're really, predator-prey doesn't really exist in the microbial world. There's no phagocytosis that's possible. There's no ability of one organism to surround and digest another. So we're talking about different rules. The trouble is the microbial ecologists try to push their stuff into the other field. And interesting here, there's probably no need for fast growth. These guys are growing, there's plenty to eat, nobody's fighting, nobody's trying to really outdo the other. It's a nice question of when bacteria need to move fast. And if you go slow enough, there's no need for motility because you don't have to move around. If you're producing stuff at a rate that's so slow, the diffusion isn't your enemy. So, and here's where all hell broke loose when Bob's organism started inventing photosynthesis and the rise of oxygen, nasty toxins, removal of the iron, and the invention of an enzyme called cytochrome oxidase was probably the most important invention for eukaryotic life because it's one of the few ways you can reduce oxygen with 1 electron and not get a toxic product. Okay, almost no way to do that. That's why oxygen's so nasty. but a much more energetic metabolism, respiratory diversity, and the rise of primitive eukaryotes after the symbiosis event. And this again was a terrible thing. Okay. And then I just want to mention one thing we talked about at lunch, that oxygen is the only electron acceptor that's a good electron acceptor in terms of energy that's a gas. If you want to develop three-dimensional life on land, you have to have an electron acceptor that's a gas. CO2 would not give you much, but oxygen gives you everything we see. So until the oxygen, you couldn't have any kind of technology unless you can figure out how to do it underwater. And you can't have really 3D life on land. Interesting stuff. Okay. I think my time's up. Oh, good grief. Okay. Take-home lessons. Prokaryotes are chemists. Okay. They're not biologists. They have small cells, high surface to volume, diverse chemistry, and no predation. But their activities leave signals. Eukaryotes are biologists. They're boring chemistry. Okay. Early life was anoxic, and oxygen changed everything. And prokaryotic life leaves its fingerprints. And I'm just going to show you one other thing, because I think this is my iron cycle that I would take out of Stuhm and Morgan's book. If it was a planet with a little oxygen in it, but no life, I don't know how you'd do that, but you would see that the iron would get oxidized here to iron 3 and form an insoluble rock. It would sink. Any of these forms could form iron hydroxide, goethite, or hematite. Eventually, if it got anoxic enough down here, you'd get some slow reduction, and you'd form a bunch of minerals. Here's what it looks like with life. We have iron-reducing bacteria speeding up this reaction by about a factor of 1,000. We have anaerobic iron oxidizers that use nitrate. It'll go this direction. We have photosynthetic anaerobic oxidizers that will run this reaction over here, the ones that Diane showed. We have iron oxidizing bacteria that will use oxygen up in the aerobic zone. We have siderophores that will bind oxygen. We have all kinds of oxygen protection mechanisms that will get rid of the toxic oxygen. And we have magnetotactic bacteria that will make magnetite inside the cells. And this is only a small portion of the way that life deals with iron. And again, this is the kind of stuff that I think we have to know if we're going to search for life elsewhere, because I claim that you can probably, these particular molecules here, organics of many kinds bound to iron, would be a dead-on biosignature. Okay, this is one case. It's not organism specific, it's life specific, I think. You don't find this without life. And furthermore, I think you could do this for virtually any elemental cycle on the planet, because the one thing that every one of these reactions has in common is that it has a bound iron in it. And that's it. Thank you.
Okay, we can have one question. That's your 4 minutes of questions.
Where do you find the electric bacteria?
Say again?
What location do you find the electric bacteria you mentioned earlier?
You'll find this organisms like the one I isolated, the Schuonella, any place there's an oxic-anoxic interface, plenty of iron, and not much sulfur. Okay, these are very hard to find in the marine environment because of all the sulfide rises up and precipitates out all the iron. So luckily I was at the University of Wisconsin, and virtually any lake in Wisconsin with an oxic-anoxic interface, you find these bugs. There's another organism called Geobacter, which Derek Lovely isolated from the Potomac River in a much more reduced zone. it lives down in the sulfate reduction zone, but there isn't much sulfate in fresh water. So he found that organism. Mine's up here at the oxyc-anoxic interface, his is down here. But really, by now, there are 100 publications about these electric bacteria. So there, I think even the biggest cynics are now believers. Okay, so